My name is Tarun, Batchu, and I'm a 7th grader from Hyatt Middle School.
Hi, my name is Shreyas, Gorthy, and I'm a 7th grader from Hyatt Middle School.
Our science fair project is testing the replication of the characteristic trait of marimo algae when asexually propagated to zooxanthellae. In this project, we are determined to find the premium ratio of marimo. Essentially, the project revolves around the question: if you breed both zooxanthellae and marimo algae, then the resulting algae will be buoyant during the day and sink at night because its parent algae, marimo, follows the same traits. After executing the given procedure assigned to this project, the prime discoveries consist of moving marimo cells, a view of the marimo cell, and the buoyancy of marimo. After asexual breeding for 2 weeks, the alternate propagation called for better results than expected. When testing the unembellished marimo, the algae is to sink, regardless of transporting the appended algae, marimo, to the ocean bed. In implementation of marimo in coral reef, we will be able to replenish reef barriers in decline, such as the Australia's Great Barrier Reef. The Great Barrier Reef has been on decline and has dropped to 7% coral cover population. Coral has been on decline due to COTS, bleaching, climate change, and cyclones. The primary objective of this project is to eliminate the COTS and bleaching factor in the reduction of coral reefs. Coral reefs have large-scale implications on the planet, even with their small surface area. They're able to generate up to half the world's oxygen and eliminate 33% of the world's carbon dioxide in the atmosphere. Even with all these benefits, coral population has been on a decline and has nearly halved in the past 30 years on account of various reasons such as climate change, coral bleaching, and COTS. The goal of this project is to ascertain an appropriate ratio to create a hybrid algae with the eventual potential to boost oxygen production. Corals are made up of animals called polyps. Polyps especially provide oxygen for the reefs. And another species that is important for coral life is algae. In more specification, zooxanthellae. Zooxanthellae is an algae that works with polyps. Zooxanthellae is a plant algae mix that works with polyps, and zooxanthellae provides oxygen and glucose, while coral polyps provide carbon dioxide and waste nutrients. This symbiotic relationship builds. In this experiment, the algae used was zooxanthellae because this is the only algae that works with coral. But the issue also stems from zooxanthellae. Since zooxanthellae is buoyant, it can't go to the ocean bed where the reefs are located. So another algae, marimo algae, was used in this propagation. Both algae were propagated and referred to as marimo. The dependent variable is how much marimo is produced, while the independent variable is the amount of marimo algae in each petri dish. I'm going to tell you a little more about these two algae here. Scientists conducted some research on marimo algae. Why do they float during the day and not at night? Was light the ultimate trigger? The truth is, day-night fluctuations are anatomically dependent for the rising and sinking of marimo algae. At day, the ball rises, and at night, the ball sinks. They also discovered that when light is introduced, let's say midday, the algae is less likely to pop up compared to if it was introduced during its entering sunrise. And this is because it's biological clock. So now moving on to the purpose and the way that we're going to conduct our process. So there's not much to do with this given information, but you might have heard more prey, more predator, or if the prey population increases, the predator population increases as well. That's not the same thing that we're doing here, but it's very similar as zooxanthellae and coral reefs stay in a mutualistic relationship. We'd like to increase coral colonies by increasing the amount of zooxanthellae in the ocean. So the life cycle of zooxanthellae consists of 2 stages, the cocoid stage and the mesomasticate stage. In the cocoid stage, the algae goes through a process where it binds with the coral and it starts a mutualistic relationship or a symbiosis. And when it has enough nutrients, it doesn't power photosynthesis. It reproduces. It goes through a process where it reproduces either asexually or sexually, or a combination of both. It goes through a process called meiosis, and that starts in the dark. And it starts in the dark, and after some point of time, it comes to the light. When the mother cell is introduced to the light, it begins cytokinesis, in which it splits up its cells and it reproduces. This new cell is now in a mesomasticate phase, and it will grow and eventually build the same relationship with coral. Shreyas is going to continue with the algae selection methodology.
While we were attempting to actually select the best algae in order to conduct our procedure, the list was very small and it was very difficult to choose, but we ended up choosing zooxanthellae and marimo algae for various reasons. Let me tell you them now. First, let's talk about zooxanthellae algae. Zooxanthellae algae is the only algae in the entire world that has a symbiotic or mutualistic relationship with coral reefs. A symbiotic relationship simply means that two animals or organisms that are working together have a positive effect on each other. But since zooxanthellae algae is actually, it cannot sink down to the ocean bed and it can only float at the ocean surface, we decided to incorporate a second type of algae into our experimentation. This algae is known as marimo algae. Marimo algae's ability to stay at the ocean surface during the day and sink down to the ocean bed at night played an important role in our experimentation. And after much thought, we decided that we were going to use marimo algae and zooxanthellae in order to create a hybrid algae that would eventually boost oxygen production.
This biological clock is known as a circadian rhythm, and marimo's circadian rhythm was very important. Shreyas will continue with the materials.
So some of the materials that you're going to need in order to conduct our experimentation are petri dish, carbon dioxide, a microscope slides, a cover glass, water, separate copper glass, zooxanthellae, marimo algae, and a pipette. So what you're going to want to do first is arrange the petri dishes and insert water, zooxanthellae, and marimo algae into them, and then insert some carbon dioxide. Then set these into a dark environment where no light can reach them for about 24 hours. After 24 hours, remove them from the dark environment and spray some carbon dioxide onto them. Make sure you spray carbon dioxide onto them every day. Then record your observations and set them in a ultraviolet light rich environment for about the next 2 to 3 weeks. After 2 to 3 weeks, trim the outermost barrier of the moss ball in your petri dish and set it in a big cup or bowl filled with water. After this, if the moss ball sinks, then you know it's marimo. If it floats, then it's your original marimo moss ball. After this, take a sample of your marimo and set it under the microscope. Observe and record your observations. Tarun will now continue with some of our background graphs.
These are our background graphs, and these supported the claim in our introduction. But without further ado, let's get to our own graphs. So I'd like to explain to you this picture, and this picture is a cell of marimo that went back getting nutrients or some sort of liquid supplement from zooxanthellae and marimo. It went from zooxanthellae to marimo, and it followed this process and it increased the volume of marimo. Now getting to the graph, this is the center of the whole project. This is what we wanted to collect it. At 0 marimo algae, there's 0 marimo. At 0.005 grams of marimo algae, there is 0.003 grams of marimo, which means 60% is converted. At 0.007 grams of marimo algae, there is 0.004 grams of marimo, which means 57% is converted. These two are our best specimens, and thus we believe that the best ratios exist between these two. At 0.009 grams of marimo algae, there is a special trait. The marimo was growing specifically on one side, which indicated that it was leaning towards one side and it was floating at one side. So it was sinking and floating, and the tug was further equalized, and it resulted in the outlier that I just mentioned above. At 0.63 grams of marimo algae, there was 0.0017 grams of marimo. This is far less than the ratio of 60% or anything. We just used a lot more marimo algae in this one. And we also noticed that as far as we increased marimo algae from here, there's no marimo produced. At 0.84 grams of marimo algae, there was no marimo produced. And furthermore, Shreyas is going to explain to you his graph and another picture.
So as you can see in this picture is representing zooxanthellae, and if you look quite closely, then you can see parts of the cell. You can see the insides of it, and this graph just simply represents the amount of marimo that was produced and was not produced. This green line represents the amount of marimo that wasn't produced, so all the algae that was still just general marimo. While this orange line represents the amount of marimo or marimo that was produced. So we're going to continue with the data analysis.
In further analyzation, coral population has declined from 27.5 to 14% of existing coral-covered population in the Great Barrier Reef, and has declined 10% totally over the north, center, and south latitudes. Now moving on to marine. The best ratios of marine are 0.005 grams to 0.007 grams of marimo algae to 12.5 milliliters of unornamented water to 12.5 milliliters of zooxanthellae. Both these specimens portrayed an active buoyancy, did not manifest buoyancy, and when observing under scope, they both showed moving cells and marine a new cell compound. Trends in the given 2-week asexual breeding process included a manure smell during day 6 through 8, and an outlier that was 0.009 grams of marine algae, and I just talked to you a little bit about that. So these two specimens we believe are the best, and this 0.005 to 0.007 grams of marine algae produce or convert the most marine. And ultimately, 3 out of 5 marine algae tested have outermost regions converted from Aegograppillinae or marine algae to marine. Shreyas will continue with the conclusion.
Throughout this project, we discovered the best ratio between both of the algae included in marine. This ratio was 0.005 to 0.007 grams of marimo algae to 12.5 milliliters of water to 12.5 milliliters of zooxanthellae algae. So while we were actually attempting to find the best ratio that I just told you about, we also discovered lots of benefits that would actually help in the real world, such as oxygen depletion, carbon dioxide emissions, krill influx and its microplastic outturn, and several other problems that our algae could help fix. The results of our project not only supported, but actually exceeded our original scientific assumptions. And the independent variable of our project was the amount of marimo algae the amount of marimo algae per petri dish, whereas the dependent variable was the amount of marine produced. The constants in our project were the amount of sunlight reaching each petri dish, the amount of water, and the amount of zooxanthellae. And the benefactor, we think that the benefactor of our project was actually setting the petri dishes in a dark environment before bringing them out into a UV light rich environment so that they could actually begin cytokinesis. If we were to do this project again, we think that we would actually increase the amount of samples as well as increase the amount of propagation time so that we could get more accurate results. Tarun, I'll continue with the applications.
There's a vast number of applications to this project, how increasing greenhouse gases can melt the polar ice caps, increase temperatures due to the growing use of fossil fuels, and many more. Although, I'd like to get into the primary applications and scientific utilities into detail. Subsiding carbon dioxide and its consequences. Coral reefs eliminate 33% of the world's carbon dioxide, and we want to increase this number even more. We want to bring this number to 50 or maybe 60%. And what happens with this is the outturn of the given action of CO2's biggest threat. CO2 or greenhouse gases have far-ranging environmental and health effects. They create respiratory diseases with smog and air pollution, and they also contribute to extreme weather, food supply disruptions, and increased wildfires with other effects caused by greenhouse gases. Oxygen depletion is corresponding escalation in temperature. With an already increasing temperature due to carbon dioxide, oxygen plays a role as well. If the oxygen levels get too faint, then there's an increase in temperature because sunlight is able to reach the Earth's surface and evaporate water vapor. Because water vapor is a greenhouse gas, temperatures rise. But with this project, we're able to eliminate any foreseen drop of O2, which prevents an increase in temperature. Oxygen scarcity is given repercussion. CO2 is something that is eliminated by corals, but corals also produce oxygen. They produce 50% of it on planet Earth. We'd like to make this number could also be further amplified. And what happens here is, the truth is, the amount of O2 we have on this planet will only last for 400 more years. And though chemicals like nitrogen, argon, or carbon dioxide may last in the atmosphere, it's unable for humans to breathe in air lacking oxygen. krill influx and its microplastic outturn. Higher oxygen levels signify animals can grow larger, healthier, and still maintain the supply of oxygen in their muscles. This complies with krill as well. And an influx of krill means more broken down plastics. A 2018 study discovered that Antarctic krill play a role in breaking down microplastics. They break down these microplastics into even smaller, non-harmful nanoplastics. Phytoplankton depletion and its marine bioeffect. By providing a glut of marine to coral reef, we are able to divert the animal from zooplankton. The animal is, like humans, it is important for sugar, supplement sugar to satisfy the obligations of coral. So it also eats by catching tiny floating animals, called zooplankton. Although without plankton or phytoplankton, there's no marine biome. And there would be no fish, and thus no fish means no food for millions of people. Without any ocean life, millions, if not billions of people will start to starve. In this project, since marine provides a supplement of starch as well, we'll be able to at least divert the coral from its consummation of zooplankton. The ultimate goal of this project is to replenish repairs and damage such with a healthy ratio of marine. The tests to execute this plan included fluorescent green light test, and we would also like to put some tests of inversion against it. In the future, we'd like to change some more things when it comes to some variables, and we'd also change the amount of like Shreyas said, the time that it takes to propagate. And if it's theoretical to further extend this procedure, we'd like to reach every single one of these applications and save the Earth as long as possible. And this is our journal. Thanks for listening to us.
Thank you.
Thank you.
All right, so this was a phenomenal project. I just had a couple questions regarding it. The first one was, what interested you to pick up this topic, and like, where did you get the idea for this topic?
What interested me about this topic is like the project itself, the implications, the never-ending reach, and the project itself. The fact that if we don't do what's right today, then the future can't live like us tomorrow. So if we can conserve the Earth and do what's best for society now, then the future can live just like us without any harm. There won't be any harm against them. So a couple morals that helped us kept me going in this project were that if you don't do what's right today, then you won't be there to do what's right tomorrow. And something saved is something earned, and something that's not bad is something good. And all these morals kept me going, and they interested me to create the project and feel good for it, or feel love and passion for the project.
Yeah, that's a wonderful passion-filled, futuristic-looking response. So going off of that, so tell me, did you find this idea published in research before, or if so, then how did you modify your idea to be specific and change and something new?
This was published before. Each algae's reproduction was previously documented, but they were never put together as marine or this component. This is completely original and only based off of the scientific aspect and inquiries of me and Shreyas. And when it comes to the applications or implications, they've all been previously established, although they've never been put in the combination that we show in this project, and they play a very important role, but they have not been put together in this combination before.
I see. So what you're telling me is that the Mary Anthony that you have produced is solely based on your project alone?
That is solely based on the inquiries of me and Shreyas.
That's amazing. My next question was, so let me hear a little bit about your project one more last time in a very simplified way. What was the question you were answering, and how did you go about answering that question?
The question that we were trying to answer is, will a bred version of zooxanthellae and marimo algae result in a hybrid algae that follows the same characteristic trait of buoyancy as marimo algae? And we went about solving this question by actually breeding both of these algae, zooxanthellae and marimo algae, breeding them, and coincidentally discovering all of these benefits.
Yeah, wow. Yeah, when I was listening to your presentation, the amount of benefits was quite enormous. Moving on with my next question, what did you learn when you were, what did you learn about science itself for your project when you were doing it before and during the experiment?
Before the project, the sciences that we were into or we looked after were the asexual breeding processes, the breeding process of marimo, the breeding process of zooxanthellae, and how these two algae work, the characteristic traits. We just looked into the algae and how do these algae behave? How are these algae going to behave if we breed those two together? And we just looked about like the algae and stuff. But throughout the project, we discovered some incidental benefits, like Shreyas said, and we understood how these could, further sprout. Like, how can one benefit actually be 5 benefits? How can one good thing be 5 good things? So we kind of sprouted our ideas. And let's say, if you talk about the krill influx that I talked to you earlier about, then that comes from oxygen. Coral reef can make the same amount of oxygen, it can do the same thing, but it can also break down plastics. So we were able to cover 2 topics with that. We were able to cover microplastics in the ocean, and we were able to cover, oxygen. So we were just sprouting our ideas, and we learned more about the sciences of environmental science, and we looked into oxygen, carbon dioxide, krill influx, and all these things.
Wow, sounds like the project was very helpful in increasing your knowledge base when it comes to science. So then tell me specifically in your experiment, like what did you learn from the data that you found?
So from the data that we were able to gather, we learned how much coral is really dying and how big coral decline really is. Which is why we created this hybrid algae. By giving it off to coral reefs, they would actually be able to reproduce, and coral reefs would actually grow faster. And since this new coral that we've developed, most of the time it stays near the ocean floor, it is able to gather with more coral more quickly, and which will actually decrease coral reproduction time by quite a bit.
I see. So tell me specifically, what was the ratio that you used to get like the maximum production of the Vera anthill?
We use 12.5 milliliters of zooxanthellae to 12.5 milliliters of water to about 0.005 grams to 0.007 grams of marimo algae.
Our best ratio was 2,500 to 1 in terms of zooxanthellae to marimo algae.
And can you tell me percentage-wise how much Vera anthill was produced from the graph?
57 to 60% are 2 best specimens.
I see. So 2 out of every 3?
Yes.
Wow, okay. Nearly, yes. Okay, so moving on, looking at a future aspect, if you had to do it again, what would you change? And then what improvements would you make from your project?
If we were to do this project again, we think that we would actually increase the breeding time between both of the algae, as well as increase the number of samples that we use, so that we could actually get more accurate results.
Okay. Yeah, and then let me hear about what's next. Are you going to continue this project, or are you going to jump to a different topic?
We're definitely going to continue this topic. We're going to have a variation. So the next variation would be how do coral reefs, or how does green galactic coral respond to the propagation of zooxanthellae and marimo algae? And in the near future, we would like to add amylase enzymes to our mixture here, because amylase enzymes convert starch to sugar. So we believe that we could have this mixture of carbohydrate become all sugar, and that would help quench coral. And we would like to apply after this in the near future. We'd like to put some more tests against it, because you want to make sure it's nice and healthy. And in specification, we'd like to put a test of inversion against it, because in the ocean, you have a lot of difference in temperature. So a test of inversion against marimo, and then we would like to see how this algae works with animals, because if we want to drop it on the ocean floor to the ocean floor, then it's going to have to pass through some animals. And coral and zooxanthellae live between jellyfish or clams or stuff like that. So we want to see how this new algae works with animals. And if that's all right, then we would really like to apply for some federal approvals to test in public places such as our intercontinental oceans. And if we do eventually harvest the amount of algae that we want to and reach all these applications, we like to put some of this algae against research to further develop algae oil, which is an alternative for fossil fuels or coal, and we like to help develop, since we want to harness some algae oil with the harvested algae, and we also want to work towards making biodegradable products or consumer goods with the algae.
I see. So, I mean, that was enormous when it came to like what you could go differently when it came to this project. Is there anything else left when it comes to how this knowledge can benefit society?
Using this knowledge, we think that society can actually participate in what we're doing, which would actually increase what we're thinking of doing, like Tarun said, biodegradable products and whatnot.
Yeah.
Yeah, if society can actually use the knowledge that we've given them and through this project, it would actually really help our environment. You know, it would not only would it help the ocean, but it would also help our atmosphere. And yeah.
We really hope we can reach all these applications here, and we're hoping for the best, and we would like to continue in the future to better develop the idea and get some more things to life and bring some more things to life. So it's been really nice talking to you.
Yeah, definitely. I look forward to these future benefits.
Thank you.
Thank you.
Thank you.